Hi, I'm Andrew. I've been invited to share some of my experience. I'm a second year Masters of Science Bioinformatics student, currently in my last semester. And I think I'll tell you a bit about my journey, my experience in the course, and also about what the project I'm working on is like. So my journey, well, it's pretty funny. It actually starts back in high school. So I come from John Monash Science School. If you don't know, that's a science school on the corner of Monash Uni Clayton campus. And at John Monash, we have a year 11 computing class. And in that class, we got to invite some academics from Monash Uni to come give us a project, and that acted as an assignment. And my group got given a bioinformatician. And we worked on our project. We did well, and we ended up being able to go present it at the 2018 Lawn Genome Conference. And needless to say, I was hooked. I just thought it was so cool using computational tools to solve complex biological problems. And naturally, I then went to Monash and did a double degree in biomed and computer science, very normal. And then it was a toss-up whether to go into an honours year or go into a master's. And ultimately, I was swayed to doing the master's because I felt it gave me a bit more of a comprehensive skill set in this stepping stone towards bioinformatics. So now I'm here. My learning experience, well, when you first get into bioinformatics, it can be a little overwhelming. There are so many different tools, algorithms, workflows, pipelines, research areas as well. They've put a little board up with them as well. It can be a little overwhelming in what to take on first or what you want to choose to do. But thankfully, in this master's, I have some really great subjects and some great teachers as well. I mean, I'm pretty sure, I think Stephen's subject should be done before doing the research project because it's so useful in the skills you're able to do. I'm currently doing Grace's subject at the moment. And I now realize I have an assignment that's starting tonight. But so far so good. And I've only heard good things from other students who have already taken that as well. But the subjects in this course really help refine down or distill the different areas into what you need to know, where you need to go in the future, because the field is moving so fast, especially with generative AI at the moment. I mean, I think I checked out a bioinformatics textbook from 10 years ago, and it was all written in Perl. So the languages are even changing faster than we know as well. And another specific highlight I wanted to point out about being in the master's course, you are heavily exposed to the Parkville precinct, so the different research institutes in the area. It's just a high density of great researchers and people doing incredible work, and you get to interact with them and do a research project, and it's all really awesome. I also wanted to talk about the community, the student communities as well. So at UniMel, they have UMSU, which is the student union. They offer a whole bunch of support services. If you're already UniMel students, you know this. But the Bioinformatics Graduate Student Association as well, they're fantastic. They host a whole bunch of events throughout the year. You get to talk to other people in the course, and you're all bonding together, maybe struggling together, but you're all in it together is the main part. So you're all fantastic people with the same interest. And there's so many different research areas, so you get exposed to different things that other people are working on as well, not necessarily related to your research project, which I might discuss a little bit about my project from a very broad aspect. It covers drug design for genetic diseases. So the idea is that by using computational tools, we want to reduce the cost and time in which it takes to make pharmaceuticals for diseases. And we want to try and understand those diseases as much as possible as well, so we can have new ways of tackling cures or preventions in treating these patients. So one of the fundamental questions you might like to ask is, how does a genetic mutation affect a protein? Whether it affects its function, its shape, how stable it is. And of course, we can use computational tools to try and predict what happens to it. So you may have a specific protein, you may have a specific mutation, feed it into an algorithm and it can tell you, yes, this makes it more stable or less stable, or it affects its function. But the main problem with the algorithms I'm looking at is that we simply don't have enough data. It's based on experimental values, which take a long time to verify and then distill into usable data sets for training these sorts of algorithms. And so one of the possible solutions we can try is simply by rotating the proteins, which I'll help illustrate with something like this. So it may look like a bit of spaghetti, but we can distill it down. The green structure is the original orientation of a protein. And the red structure is the rotated version. So you can see a few of the different amino acid groups pointed out, rotating around. And this cube is what is input into the algorithm that is being trained. And so it's within this cube that the algorithm calculates different chemical and physical properties to try and make its prediction. The little blue ball in the center is the target amino acid which can be mutated. I'm pretty sure in this one it's a glycine, and so it doesn't really have a functional group, but you can see the difference. See, there's a tyrosine that pops in and out of this little capture area. So it's trying to help illustrate that these are new data points we can use and that they are reliably different in being able to be used as a new data point. So this is just going around the x-axis. This one is going around the y-axis. I wonder if you can see another tyrosine sort of pop into frame and then pop out of frame, as well as phenylalanine scrolling down the bottom. So these are usable different data points that can be used to help improve our algorithms and try and make them better predictors in whatever we're trying to calculate. And if you're good with motion sickness, this is going around the x and y axis. And so it can become very, very different. And the hope is that we can make better predictors by just generating new data from what we already have. And just a quick reflection, in the degree, I've developed new skills, I've made new friends, and I'm a bit biased, but I'm very excited about the future of bioinformatics.